So I'd like to take a moment to talk to you about a novel approach to cell therapy. You may know MaxCyte as an electroporation technology company, and we do have two divisions of our company. One is the instrumentation company, and one is the cell therapy drug development group. I have to post a forward-looking statement because we are public on the London Stock Exchange. So our company as a whole was founded in the late 90s, and in 2016 we implemented a new business unit. As I mentioned, the first business unit and the long-standing business unit is our instrumentation flow electroporation side of the company. And in 2016, we raised money to start our own cell therapy drug development programs internally. All of the products that I'll talk to you about are protected by global intellectual property. We're based outside of Washington, D.C. in Gaithersburg, Maryland. And as I mentioned, we're public on the London Stock Exchange. Oh, going the wrong way, sorry. All right. So just a visual of the company, because I do get asked a lot of questions about the instrument side of the business, of which I'm not very adept at discussing. However, just to visually explain that we do have a cell therapy drug development division. We have a proprietary platform that we develop our CAR therapies from, and we also have a product in the clinic. Oh, I keep going the wrong way. Apologize. All right, so our platform, which is proprietary to us, and no one else creates CARs of this type of construct, we use mRNA as our CAR, no viral components, and we transfect the mRNA into fresh PBMCs. Almost all other CARs are CAR-Ts, so you won't hear me describe our products as CAR-Ts. And companies use other isolated and expanded cell types. PBMCs, as you know, are made up of 4 different cell types, and that's the basis of our autologous cell therapies. A little bit more about our platform attributes is excuse me, these lights kind of hard to see. We manufacture our product in less than a day. So for autologous cell therapy, we are in a very unique space. Our product takes about, from the time the apheresis is drawn from the patient to cryopreservation of the doses for the patient is about 17 hours, which means our turnaround to the patient is much more rapid than any other CAR-T autologous product. We purposely constructed our CARs such that there's transient expression. In other words, we want to control the potential for toxicities with our CARs. And therefore, using the mRNA, we can control the persistence once you put that product into a patient. We're very proud of our manufacturing process, which, as you've probably heard from many other CAR companies, manufacturing is 80% of what you do with a successful product. We're GMP process that is qualified, and our feasibility has been completed. We are in the clinic, so the FDA, as our regulatory body for our clinical trials, has allowed us to move forward into human studies. We do use the flow electroporation instrumentation from our base business to transfect the mRNA into our cells because the transfection rate is so high, we don't need to expand or activate those cells prior to putting them into patients. I've already mentioned from apheresis to cryopreservation is less than a day, and turnaround to the patient is really less than half of other autologous cell therapy products. In one apheresis, the efficiency of our manufacturing process is so high, we actually can generate 10 to 100 doses per apheresis, one apheresis of a patient. This is important because we are a multi-dosed product, and I'll explain our hypothesis, so to speak, of our impact on the immune system. Just a side-by-side comparison of CAR-MA, which is what we call our CAR platform, and other CARs. Our turnaround time is extremely rapid back to the patient. We don't have a viral component, so obviously that reduced cost of goods of our product. We, as I mentioned, employ PBMCs as our API, where other CARs are employing a single cell type. The benefit with PBMCs is you not only get T cells, like CAR Ts, but we also are employing NK cells, which we all know also have that beneficial impact on the immune system for cell therapy. And our multi-dosing opportunity, because of the efficiency of manufacturing in generating more than one dose out of one patient apheresis. Just a quick review of our pipeline, little snapshot. Our first product, MCY-M11, is intraperitoneally administered as a proof of concept study to show that our product actually can get into the cells and the cancer lesions for these patients. As you know, ovarian cancer and peritoneal mesothelioma are pretty much confined to the peritoneal space. So they're ideal candidates for this IP study. We will also take the exact same drug and administer it IV in some subsequent studies we have upcoming. Those will be in different tumor types, and I'll explain our opportunity to go into multiple types of solid tumors with the same product in a moment. MCY-C55 is a CD123 targeted product for AML, and that's being worked on in animal studies, IND enabling. Just a snapshot of our construct. We don't look a lot different from CAR Ts, third generation CAR Ts, just to point out that our co-stimulatory domain is 4-1BB. So mRNA, how long does it last in our PBMCs? So we took 3 groups of donor cells, transfected with our CAR, and we found over 7 days, maybe a few more than that, we still had functional activity of the cells, and there were still some cells hanging around. So that gave us the basis for designing a protocol for dosing our patients in the clinic. And what's our postulate for the immune response? So unlike one and done CAR Ts, where the patient receives one dose and the immune system takes off in hopefully mostly positive ways, there are also those toxicity issues. So we're taking a gentler approach to the immune system with the first dose breaking the immune tolerance. Second dose, reactivating the immune system. And the third dose generating that immune cascade, which we all desire with these cell therapy products. Now, in our original studies, and I'll get into this in a moment with detail, we had 3 doses, and we have 3 doses we're treating our patients with in the IP study. We have additional data just recently over the Easter holiday to show that we can dose patients further and have a better overall response rate in animals, of course, at this point. Why did we choose mesothelin? We get this question all the time. Most small companies like us start with CD19, BCMA. We really felt that our approach to the immune system would actually have a benefit in solid tumors. So we chose mesothelin as our target. Mesothelin is expressed in over 200 solid tumors. As a small company, it gives you an enormous number of tumors that you can consider. As well, our business model is to out-license our products. So for our potential partners in the future, for our mesothelin product, it also gives them a multitude of tumors to consider outside of the ones that we are studying. So animal data. We dosed the animals with ID8 cells, which are peritoneal cancer cells. And as you can see, the mice are growing tumors very well on the bottom part of the slide. And from controls, which are the last 3 doses, to our lowest dose and our highest dose, you can see why we chose the higher dose. The tumor debulking of the animals was much more impactful. We took that high dose and took it into overall survival models, same animals, and nude mice. And you can see in the black line is the control, where the animals all died. There was no survival. 1 dose, and at 3 doses, which is the green line, you see an overall survival of 20%. Not terribly impressive. We recently received a data analysis on animals dosed out with 6 doses, and I don't have that slide because it just came out, and I didn't want to misrepresent it visually, so I'm just speaking to it. At 6 doses, we had a 50% overall survival rate, which is very encouraging for us to now use that as data to move forward in our IV study with 6 doses in the patients. So as I mentioned, our current study is an intraperitoneally administered MCY-M11. The tumors are ovarian and peritoneal mesothelioma. Peritoneal mesothelioma is a very rare tumor. We have the one site in the United States that treats over 90% of those patients, the NIH, as an investigative site. Ovarian cancer is a very, very high expressor of mesothelin, so great opportunity to bring those patients into the study as well. And if you know anything about ovarian cancer, there is an enormous unmet medical need with those patients post-chemotherapy. There really isn't much left for them. So perfect candidates for mesothelin-targeted CAR. Our trial sites, WashU St. Louis, specifically just enrolling ovarian cancer, and the NIH, we have 2 investigators. Rafid Hassan, who's the global expert in mesothelin, is our investigator for peritoneal mesothelioma, and then we have a different investigator for ovarian cancer. Essentially, it's like having 3 sites, which is kind of a bonus for us as a small company. Each patient will receive 3 doses because that's the animal data that we had to submit to the FDA to design our study initially. And then, as I mentioned, now we have the 6 doses for animals, and we'll move that into our IV study. And we are open and enrolling. I can't say how many patients at this point in time, since that's not been publicly disclosed, but it's very exciting for us to be in the clinic when we started this cell therapy group in late 2016. So we've worked very quickly, very efficiently to move our products and our efforts forward. Our business model, as I mentioned earlier, is to out-license our products. So we are out-licensing the platform for other targets that we're not initiating in our own pipeline. We're also seeking partners for M11, our mesothelin-targeted CAR in the clinic, and also seeking collaborators for new targets to populate our pipeline even further. Just a snapshot of our leadership. We're very proud of the subject matter experts that have joined the MaxCyte Karma cell therapy team. Decades of experience, which is critical for a small company, and I think you've probably heard this over and over from other cell therapy companies presenting today. It's imperative. There aren't a lot of folks running around with cell therapy experience, so for us to be able to garner those experts have been a testament to the work that we're doing and the science behind the work that we're doing. Thank you very much.